Welcome back to Science Her Way, everybody. Today we have a fascinating episode featuring Dr. Sarah Oppelt, a distinguished scientist and educator specializing in molecular biology and biochemistry. Dr. Oppelt has a PhD in molecular cell biology and biochemistry from Boston University and a BA in biology from Hartwick College.
So Dr. Oppelt's work focuses on how metabolism influences cell fate, and she's currently researching the impact of dyslipidemia on metabolic distress and inflammation. But before we dive into the science, let's welcome Dr. Oppelt onto the show. Thank you for joining us.
Guys, thank you so much for having me. And it's very rare that people refer to me as Dr. Oppelt. Usually people call me Sarah. If we need to be formal, you can call me Dr. Sarah, but okay. Sarah's just fine with me.
Yeah, perfect.
You can do Dr. Sarah. That's easier.
Yeah.
So we don't butcher the last name again.
No, you guys are doing a great job.
All right.
So we are so thrilled to have you. We always like to start talking a little bit about the journey that got you where you are now. So what led you to science? What did you decide to pursue a career as a female scientist? So tell us everything.
Yeah, absolutely. So as somebody born with 2 X chromosomes, I didn't have the choice about being a female scientist. Like, I just...
Yep.
But yeah, I mean, this is really one of those things. I think there's a little bit of a gift and a curse, because I was just like born being very nerdy and being very interested in how the world around us worked, and especially how our body worked. And when I was younger, there's no other scientists in my family. So if you were nerdy and if you liked, anything that had to do with bodies, everybody thought you were going to go to medical school. So when I was in college, I thought the same thing. I thought I would go to medical school one day. Mm-hmm. And it became clear through my undergraduate career that much better suited for research and some of those open-ended questions. And it was my undergraduate research advisor who was just like, hey, the grad school is an option. And I was like, no, I did not know that. And so, so Dr. Stan Sessions at Hartwick College really pointed me towards being a research scientist as an option that I can do. I ended up getting my PhD from Boston University in Dr. Dean Tolan's lab, and now I'm here.
Yeah, that's really nice.
So what drew you to the field of biochemistry? It's definitely an area I'm passionate about too, but I'm curious, like, from a nutritional perspective, but then...
Yeah. It was a complete accident.
Okay.
So for everybody who doesn't know a biochemist, biochemistry is really the study of proteins. And there's many different ways that people can be biochemists. So some biochemists will, strictly talk about like bonds that are changing in different, active sites of enzymes or whatever. What I personally do and what drew me into biochemistry is that I really love how systems work. Okay. So I thought when I got to grad school that I would end up studying something like developmental biology and looking at how different pieces came together as a dynamic system to create a new phenomenon, right? And with developmental biology, that's how you get different gene expressions leading to an entire organism. Mm-hmm. And Boston University, when you first come into grad school, you'll do rotations through different people's labs to make sure not just the thing that you think is interesting is actually interesting to you, but how that science is done on a daily basis is also interesting to you, because as you guys know, it's the labor of love being in the lab, mm-hmm. especially when you first get started. So I had a biochemistry class for the very first time when I got into grad school with the person who ended up becoming my PhD mentor, Dr. Dean Tolan, and it was a real slap in the face because I had never had a biochemistry class before. Mm-hmm. It's a completely different way of thinking about things, and it really opened my eyes to how the different molecules inside our cells are also organized into different systems, and the interaction between those molecules and the enzymes inside our cells determines what happens inside a cell. Mm-hmm. And so, not to be too philosophical about it, but the levels of different metabolites essentially determine whether or not we feel healthy or whether or not we feel stressed out. And I just thought that was super fascinating, how we have these complex phenomenon like stress or health, well-being, and there is a physical determinant process that we can understand a little bit better, and then it literally influences how we feel. So I just thought that was super fascinating. I also thought that the brain was like the most interesting organ, and it happened that that Dr. Tolan had a postdoc who was finishing up her time in the lab and was working on a brain metabolism project. And so he was like, oh, is this something that you might be interested in? And I was like, I will fight somebody else who says they're interested in this. Like, I want to study brain metabolism. This is what I'm here to do. And so it really, because of my interest in some of these higher level phenomenon, and because there happened to be a project open studying sugar metabolism in the brain, I then had to learn how to study enzymes and become a biochemist so that I could get to the things that I really wanted to work on.
Okay.
Yeah, that's really cool. You mentioned, and we'll probably talk a little bit about your paper that is related to gene expression. Am I correct?
Yeah, specifically expression of enzymes that are involved in fructose metabolism in the brain.
Okay, so could you just give us an overview for our listeners that are not too familiar with gene expression? So what is that? And how is that usually measured in research? Because I'm curious about that as well.
Yeah, absolutely. That is a really great question. So genes, I think I'll just remind everybody, I'm sure your listeners know, that all of our DNA, our nucleic acid sequences, are arranged into essentially two groups. And some of those groups tell the cell how to organize itself, and some of those nucleic acid sequences actually become proteins. So when we say gene expression, we're usually talking about whether or not we see the final product of a gene inside a cell. And with metabolism, most of the time, this is looking at different protein levels, but it can be looking at other nucleic acid sequences that regulate those protein levels, like messenger RNA, or the state the DNA is being stored in, how tightly it's stored or how loosely it's stored. So for me, most of the time, we're talking about which proteins you see inside a cell and what level those proteins are found at. What proportion of the cell is one protein versus another protein.
Perfect.
Does that make sense?
Yeah, totally makes sense. And I'm very fascinated about the topic recently because I never worked with that molecular level structure. I always, my background is on clinical research and what I was seeing in patients and what was going on with their function. So I'm always fascinated. And recently I read the book from the Nobel Prize winner, Jennifer Doudna, and she won the Nobel Prize on gene expression, right? She studies gene expression and how to edit genes and all that aspect. And I was really fascinated about it.
Are you talking specifically about her book on CRISPR?
Yeah, I think it's her biography, right? On the...
Yeah, she may have written more than one book, so I don't want to take any credit away from Dr. Doudna. But yeah, she started, she still is a molecular biologist who looks at gene expression and especially how bacteria regulate their ability to fend off of viruses. And so for anybody who's like curious or wants to do more reading after this podcast, if you have not heard about CRISPR in the news, she's a fascinating person to look up. Like Dr. Fair said, she recently won the Nobel Prize with some collaborators for her work on the CRISPR-Cas9 system.
It's fascinating to hear your story. As a female scientist, what are some barriers or challenges that you maybe faced along the way?
Yeah, I think that's a really great question. And I was lucky enough to be invited to talk about this with some different groups over the years. And for me personally, I was very lucky to have some strong female role models in my life. So nobody else was like a career scientist. That was definitely new for me. But seeing women in the professional workforce be ambitious was something that was in my family. And my immediate parents were always really supportive of that. And when I was really young, I had the chance to live with my great-grandparents, and, they were a little bit older. So when it was very clear that I was interested in science and I was interested in how the body works, they would always joke around with me that, they were getting older and they needed a doctor, so they were in Metabolism, Massachusetts. So they were like, oh, you have to come to Massachusetts and become a doctor. And so when I was really little, I know that this was a joke for them because they were just old people.
Yeah.
But for me, I was like, no, I promised my great-grandparents that I would move to Massachusetts and become a doctor. So I was very fortunate that they passed away in their 90s when I was around in my 20s. So I didn't have to go to medical school, but I did promise them that I would go to Massachusetts and become a doctor. So I did have that support growing up as a younger person. I also, I kind of had that personality where when I've made up my mind to do something, I don't necessarily see that other people are discouraging me from it. Mm-hmm. So there isn't necessarily a time in my professional life where there has been clear sexism or clear barriers against women. There's definitely some microaggressions that I think we all face that can be hard to identify in the moment.
Sure.
Now as I'm established in my career, I'll talk with friends about are there lactation rooms at the place you work? And it's just
right.
You know what?
Yeah.
That's definitely something to think about. But that didn't affect me earlier in my career. So I think for me, one of the harder things that I definitely did notice was other people's internalization of gender norms and gender stereotypes. And so I did meet a number of people throughout my life who had this idea that being feminine and being ambitious and driven didn't necessarily go together. And I got social pushback from that.
Sure.
You just decide not to be friends with those people.
Yeah.
So when I was younger, that definitely was something that socially took a toll on me. And I did have to actively say oh, this person is wrong about femininity. There's nothing wrong with me being smart or with me saying that I know the things that I actually know. Mm-hmm. And I think currently in my life, sometimes there'll be something that comes up, and I know not all women feel this or internalize this the same way, but I'll see people, especially feminine identifying people, have a harder time being confident and just very succinctly and directly saying what it is they're good at without apologizing for it. And this has nothing to do with being a scientist, but I was over the summer, I was with a friend and there was a parallel parking space that was really tight. And I was do you think my car can fit? And she was well, if you're good at parallel parking. I was yeah, I am. And she was wow, you're so confident. I was yeah, but I'm good at parallel parking. Why would I, And so then it was kind of well, do you come out and say that whenever you're good at something? And I'm yeah, I do. Mm-hmm. And it isn't always met, sometimes it catches people off guard to hear a woman just be no, I am the expert on this.
Yes.
Yes. And it is okay to say that. Yeah. So in all, career-wise, overall, very, very lucky to be supported along the way by my family and my mentors. Definitely recognize that there are some social things that we have to take a deep breath and just be no, I'm good at this.
Yeah, I agree.
That's awesome. Yeah, cool. And Dr. Sarah works with animal models. So usually gene expression, it's really common to analyze those outcomes in animal models. Can you talk a little bit about how is that work, how basic research and animal research in particular works? Because I think you are our first guest that is that has that expertise on basic research. So I think that's awesome for us to kind of differentiate the advantage, maybe disadvantages, and what we can learn from animal model research.
Yeah, that's awesome. So my work as a biochemist, and specifically as somebody who studies metabolism, does lead me to work definitely with purified proteins sometimes, but then we want to see how those proteins come together to influence what's going on inside a cell. So we have some cell cultures that we have growing in lab right now. And then also how those cells all work together to form a complete organism. So that's where the animal models come in. And there are some things that we just can't learn if our model is too reductionist, right? So, if we're doing something with purified proteins, we might be able to say something like, these two proteins have the ability to interact together, and then you can do some biochemical assays to measure their affinity for each other, so how attracted they are, how likely they are to come together in a complex, so like how much do you need before they start interacting, how strong is the bond between them, stuff like that. You don't know what effect that's going to have in a cell if you don't then look at what happens in cells. So this is when we start moving in cell culture. Now, with cell culture, we will look at one cell type at a time usually. There are some co-culture models where you can see how two types of cells interact. But for the most part, if you want to know how two types of cells interact to become an organ or to function where we move from metabolism into physiology, then you have to use animal models. And so I do at this point, since you guys are physiologists by training or nutritionists by training, I want to clarify that as a biochemist or somebody who does more molecular research, when we say metabolism, we're talking about those protein interactions inside a cell. I think that most people, when they talk about metabolism, are usually referring to like how fast we eat snacks, which definitely is a part of metabolism. I'll try and differentiate when I mean like these proteins working together in a pathway versus what our body is doing on that physiological level. So just as we listen to this, I'll try and keep those vocabulary straight for us because I know that other people use metabolism in a slightly different way.
Yeah, but continue on your more specific line of research on basic research and animal model. I think Kelly and I often talk about how we wish on a sample size perspective that we worked with animal models because they will be there always. We don't have to chase them. I think I shared with Kelly, like during one of my PhD research, on one of my projects, I had to go to, 20 or 25 patients' houses to collect blood sample because they would not come to the clinic because I planned it really poorly and it was a Sunday and they said like, I will not be coming in. And I said, yeah, but okay, what if I go to your place? It's like, yeah, sure. So I was kind of drive through between their places and collecting blood samples. So animal model has that advantage, right? So can you talk a little bit about how was maybe your first experiences working with animal model research, maybe a few of the skills that you learned with that, and I think most importantly, what is the relevance of animal model and basic research towards a more broad aspects of health sciences, how that contributes for treatments, for our knowledge, for the patients and general population?
Yeah. So first, Anna, I do want to acknowledge that your animal models will always be there for you, even on Sundays. So one of the first things that I'll do with students is be like, okay, let's talk about when you want this assay to end. Like, when are you actually going to collect your data? Mm-hmm. Because I know that you're free to set everything up on Tuesday. That means you're going to have to come back here on Sunday. And so, on one hand, luckily, yes, we can come back to lab on Sunday. But I think that is one of the things that comes with wisdom is you design your experiments to work backwards and be like, I don't want to come back for the mice. I don't want to come back for the people. I don't want to come back for the cells. So let me work my experiment backwards so I'm not doing that on Sunday. But that's just a piece of advice for everybody listening who's thinking about research design. To your question about animal models, there's a responsibility that we really do have to take when we are working with other living organisms. Mm-hmm. And so I take that very seriously, and I really do appreciate that we have this tool. So when we design experiments, my team personally, we try and take like everything that we can. This does mean that you have to think about what things you might do with the tissues in the future, because that will affect the decisions that you make about how you store them and how long different molecules will be stable for under different conditions. Mm-hmm. And so there's a little bit of a plus minus for different technical reasons, depending on what you think that you want to look at and what decisions you make so that you can get the most out of those samples. And it also means that you have to be familiar with the different organs that you're working with because, for instance, when I was doing projects with the brain and then later on with the liver, those are both pretty stable organs that for what we were looking at, you can take them out, get them on ice quickly, and kind of move on. If you're working with an organ like the pancreas, the way that you're handling the animal before you start a dissection will influence which hormones it releases, and then those hormones will have an impact on some of the metabolites you might be trying to look at. And so you have to really know the system that you're working in so that when you handle these animals and when you handle the tissues, you can treat them appropriately so you don't end up with artifacts in your experiment. It's very important that you plan out any animal experiment with like keeping humanity in mind. Like, and I know that the word is humanity, but like extending that to the animals that we work with. Because you can't be careless about those things. That being said, there's no getting around the fact that when we do want to introduce a treatment or a therapeutic into clinical studies, you have to have a certain amount of animal studies done. You have to show that what you're introducing, what you're proposing to introduce into a human population will be effective and is not likely to be toxic. So we do have to use animal models for those things. I recognize that not every scientist is going to choose that, and I think that that's totally fair. It's kind of like, it was uncomfortable for me to start working with animal models, but the advantage, I would say, is like, number one, like we have to have those if we want to make any clinical progress. And then number two, it did give me an appreciation for all of the organs inside of our body, because it is one of those few times where you get to see inside. And when I think about things, like I'm a very visual person, and because I've done so many dissections, like when I teach anatomy and physiology or when I teach cell biology, I can think about where all those cells are and which ones are touching each other and how that's going to impact communication inside the body, which is not something I would have as strong a grasp on if I wasn't doing so many dissections.
Yep. Yeah, that's awesome.
So what are you working on? I know you're doing some stuff with ultrasound. Is that correct?
Yeah, that is correct.
Okay.
Yeah, absolutely. So here at Central, we have a couple of different projects going on, and they all center around metabolism, and specifically that line between metabolic balance, where we feel healthy, and metabolic stress, where we're at risk of developing different diseases. So the project that you just mentioned, we are working on with a student here, Rhodes.
Yes.
They just presented at Source. They had a poster about it, and I am very proud of them. So what Rhodes was originally interested in was more of the physiology side.
Right.
So when they said that they were interested in student research, they came to talk to me about some of the things I do as somebody who studies sugar metabolism. And we were talking about where that line between like health and metabolic stress was. And one of the ways that you can measure metabolic health versus stress is by looking at how people use energy sources during exercise. So Rhodes's project is looking at how easy it is for an individual to switch between burning fat for energy during exercise versus burning glucose for energy during exercise. And under quote unquote ideal situations, we would like our bodies to be able to switch between those two fuel sources. So for anybody who is listening who isn't necessarily familiar with this, we store energy in our bodies in a couple of different ways. One of them is fat tissue, and then the other one is glycogen. And so glycogen is sugar chains or glucose chains, and we have some glycogen in our liver, and we have some glycogen in our muscles. And the amount of glycogen that we have varies based on a number of different factors. Once our glycogen stores are full, we then start to store things in our fat cells. So it's totally normal, and it's very necessary and very healthy to have a certain amount of body fat, because you need it for energy. We tap into our fat stores whenever we're fasting. And I know that like fast can be a little bit of a buzzword right now with like intermittent fasting or like different diets. So when we mean fasting, as scientists, we usually mean any period longer than maybe about 8 hours where we're not taking in food. So you should be thinking to yourself, that includes sleeping overnight. And if you just said to yourself, I don't ever plan 8 hours for sleep, that's a different conversation. I know because I'm a sleep scientist soon.
Yeah.
But so in order for us to have enough energy to wake up in the morning, we have to be able to burn some fat for energy.
Yep.
And then when we do other things throughout our day, like Kelly, I know that you run marathons. I like running marathons.
Yep.
I do not, people. I'm the only one. I do not.
There's no, that was not shade on like not running marathons. If it's not for you, you don't have to do it. But I just brought that up to say that there's a number of situations where we are fasting from intaking food or from using glucose as a sole energy source that we might not be aware of. And so even if you don't think that you fast during the day, you absolutely do. And you have to be able to tap into your fat stores for energy.
Sure.
Okay,
Yeah.
so that whole little preamble was to hopefully impress upon our listeners how important it is to be able to switch between using just glucose for energy and using fat for energy. So Rose's study was doing a bike exercise test to have a measure of if people were burning fat versus glucose at different energy intensity levels. And then they were also using an ultrasound to look at the echogenicity of somebody's liver. So for those of us who don't regularly use ultrasounds, which was me until a few months ago, the ultrasound image will bounce back depending on the density of whatever it is you're looking at.
Yeah.
So in the liver, it should be pretty squishy, and you should kind of be able to see through it. The more fat or fibrosis that we find in the liver, the more it's going to bounce back and have a higher echogenicity store. Now, one of the things that is associated with liver, too much fat being stored in the liver and the buildup of scar tissue or fibrosis in the liver, that is oftentimes associated with insulin resistance and the development of type 2 diabetes, which also means that as people become more insulin insensitive or insulin resistant, they have higher insulin, like baseline insulin levels just at normal times.
Right.
And in addition to insulin helping us make sure our blood glucose levels don't get too high, it also stops us from releasing energy. Insulin is a growth factor, and so it encourages our body to store energy, and that's very appropriate after meals, and yeah, it's important for normal health. But it's also important that we don't have too much of it around, because when we do fast and we do need to tap into our fat stores, we have to be able to release that energy. We also have to be able to release the fat and the glycogen from our liver. So if there's too much insulin around, we can't get at that stored fat or the stored glycogen in our liver. So Rhodes and I were asking the question, could we use liver echogenicity as kind of a proxy for whether or not somebody may be at risk of not being able to access their fat stores? And if that was the case, would that show up on an exercise test?
Okay.
Yeah.
Was the sample for physical active individuals? Were they active?
So we were recruiting individuals who were, and we still are doing some recruitment. If anybody listening is interested, you can reach out to myself or to Rhodes. We're recruiting individuals between 18 and 55 of all body types and activity. We do ask people to self-identify about how athletic they are.
Okay.
We measure height and weight, since BMI is used so often clinically as a measure of health. Because the flip side of this is somebody can maybe have like more body fat or a higher BMI, but still be very metabolically flexible,
Right.
where they can, where they can use their fat stores. And we think it's just as important that people understand you do need a certain amount of body fat to be healthy. So we wanted to see if we could separate out, was there a difference in metabolic health if we looked at BMI versus if we looked at liver echogenicity. So we didn't We didn't screen for people. We didn't recruit only people who were active, and we didn't exclude people who were active.
Okay.
It really was trying to be like a sample of the general population.
Gotcha.
Yeah.
Mm-hmm.
I think it would be interesting if you had, you know, like someone who's chronically trained is obviously going to be a bit better at utilizing fat as a substrate during exercise. So
we are hoping to get a few more samples, and I don't want to talk about any preliminary results. I know.
Spoiler?
Like, I really think that there's some stuff there that can be published, and I don't want to like put it out there before we finished analysis. But definitely more to come. A little cliffhanger.
Are you guys also checking for maybe differences among males and females to see? Okay, that will be really interesting.
Yeah, it will be. And I don't want to put words in your mouth, but I'm guessing that you asked the question because it is known that females are actually better at utilizing their fat stores. Yeah, that is something that nobody told me when I was younger, that you actually as a female might be carrying a little bit more fat on your hips because it makes you healthier.
Yep.
So even though that's not my current research as a biochemist, I do think it's a really important message for us to hear that we need some body fat.
Yep.
And just because we're holding on to body fat, that is not a sign that we're unhealthy. And I will save the rest of it for a discussion, maybe particularly on this study in the future when we wrapped it up.
Dr. Sarah will have to come back to talk about that after public. So let's talk a little bit about some research that is already published, right?
Yeah.
So Dr. Sarah just published a paper on Brain Research Journal. Is that it? In 2017? Is that correct? Yeah. And Dr. Sarah mentioned that we might chat a little bit about specific regions of the brain that are capable of fructose metabolism. So that seems really interesting. Can you talk a little bit? What was the research question and what did you guys do?
Yeah, absolutely. So like I mentioned earlier, my PhD research was on different types of sugar metabolism. So there are a number of different types of sugar. I think that most people have heard of glucose and have heard of fructose. So as scientists, when we talk about sugar, we're talking about a specific class of carbohydrate molecules. And I think that at this point, most people have heard about fructose casually, especially in the context of high fructose corn syrup. So I will give people... There will not be a quiz at the end of this podcast. I will give people a very brief overview of some of the biochemistry. For different reasons, glucose is the most common sugar that's just found in nature. Mm-hmm. So whenever we eat food and we talk about growing, we need some source of carbon, and usually that comes from glucose. There are other sugars that are made in small quantities. And so fructose is a sugar that's only found naturally in fruit. Now that we have the ability to process food, we can get fructose found in other places. But it's named fructose because it naturally occurs in fruit. It's not the main sugar in fruit, it's a small sugar. So if we were eating unprocessed diets like we did for most of our evolutionary history, our bodies would not come in contact with that much fructose. And when we did eat something with fructose in it, this was usually a sign that there was a lot of fruit around, right? Mm-hmm. And so for populations that lived in places that relied on seasonal fruit, we would need a different way for our body to sense, oh, hey, like it's harvest season, there's a lot of fruit around. Mm-hmm. And that means that our body is going to treat that sugar that we don't see very often naturally differently than a sugar we see all of the time. So when we think about, or at least like when I think about energy coming into our body, I will think about it a little bit the same way that water flows through a system, where if you have a large stream of energy or of water coming in and it's going to be constant, you want that to be regulated because otherwise you're going to have floods and that will be damaging. Mm-hmm. If you have a little bit of energy coming in, that's not going to change the overall flow of things. You don't need to spend time regulating it. Mm-hmm. So our body has very tight control, or it should have very tight control over our glucose levels. both in our blood glucose and inside our cell. When we eat fructose, if we were eating unprocessed food, it would be a very small, like 1 to, for most people, like 2 to 5%, but like maybe 1 to 10% of an unprocessed diet, right? So that's not enough that our body would spend energy regulating that type of sugar metabolism. So once it gets inside a cell, our body acts as if it's not going to contribute that much to the overall amount of energy, right? Now, once we start getting into processed food, it can be a much bigger part of people's diets, like 10 to 20% of the calories. And now we're talking about an amount of like a specific carbon source that can change our overall energy flow, which means that our cell it's now hit with like droughts and flash floods. And that is very hard to maintain homeostasis or to maintain non-stressful conditions when you have these wild swings between, oh my gosh, there's like all this carbon and all this energy we have to deal with. And like, okay, now it's all gone, but we just set up all these systems to regulate it more. And so like swinging back and forth like that is stressful on our cells. Okay, so that being said, not every organ inside our body can actually break down fructose. So the ones that can, we see a lot of stress on those organs. So like our small intestines and especially our liver. And this is where this is one of the places that non-alcoholic fatty disease can come from. You have unregulated metabolism because we have a sugar that our body thinks we're only going to see a little bit of it. So when we see a lot of it, we scramble to figure out what to do. And it leads to, in the liver, non-alcoholic fatty liver disease. And in the gut, it can contribute to IBS, it can contribute to leaky gut syndrome, so on and so forth. Together, the small intestine and the liver will metabolize, it's thought that they metabolize about 70% of the total fructose you eat. So when you talk to people, and they'll say usually like, oh, the liver is the organ that does most of our fructose metabolism, which is true. It's over 50%, but it's not 100%, right? So where's the rest of the fructose going? Number one. And then number two, is fructose metabolism causing the same amount of stress in these other organs that it's causing in the liver, right? Because if it's not, then maybe we can have some intervention that just like gets these other organs to metabolize fructose and we can take stress off the liver. Yeah? If it is causing the same amount of stress, we need to know about that because we need to think about like where in our body we're actually going to see this stress. Okay, so the paper that you just mentioned, the 2017 paper on specific regions of the brain being able to metabolize fructose, came about to, number one, try to answer the question, where else is fructose going in the body? So this started with a previous member of the lab I was working in, so in Dean Tolan's lab, Dr. Vince Finari had, guys, this was before bioinformatics was a big deal. So this was like a little bit of time ago. But Vince had written some code to mine some databases where scientists, like anybody who had done an NIH experiment using NIH money, would put their gene expression results into these publicly available databases. And if you could understand computers well enough in the late 90s, early 2000s, which I know for some of our listeners, you're like, wow, hey, before computers. This was like a big deal at the time. And you really needed like specialized knowledge and like an interest in this. So Vince really had a lot of forethought to be like, okay, I'm going to put together a computer program that goes through all of these databases to say, where are people reporting these genes being expressed in the first place? And so he made those bioinformatic predictions about where we would find the enzymes involved in fructose metabolism. And we made a short list of organs that had like the next highest expression after liver, small intestine, and kidneys, because those are the three organs that we already knew participate in fructose metabolism, and they participate in that order. The liver does the majority of our fructose metabolism. The small intestine takes care of like 5 to 10% of fructose metabolism. And then our kidneys will do a little bit also, especially as they filter our blood. So what organ was in number 4? And it turned out the brain was predicted to have these enzymes. Yeah.
Interesting.
Cool.
Yeah.
Yeah, it is interesting.
Cool.
So from there, like, a prediction is great, but you have to figure out if it's true or not. So the databases that Vince had mined were looking specifically for mRNA expression, messenger RNA. Just because you predict it doesn't mean it's there. So the first thing that we did was take sections of brains of mice and see if we could find the mRNA if the prediction was true or not. And so we did. We found the mRNAs. And then that does not guarantee that they make proteins. So then we had to look for the proteins. And we found the proteins. That does not guarantee that you have metabolism going on.
Right. Yeah.
So we then had to say, like, okay, like, the proteins are there. Can we actually see if we take specific brain regions that have these mRNAs and have these proteins, will they take fructose and turn it into carbon dioxide? That's the final measure of metabolism. And they do.
Okay.
And one of the really interesting things was that not every cell in the brain expresses these enzymes. So it's very specific regions of the brain. And this was like a super interesting finding. This was towards the end of my PhD where we published this. And at that point, people were starting, like, more and more of the scientific and medical community were starting to talk about how high fructose in our diets may be leading to inflammation. And so as I finished my PhD, I was like, oh my God, maybe there's inflammation in the brain from too much sugar metabolism. Gotta go. I know, it was both, like, super exciting because it was a thing that people had not reported on before. But it has implications for, you know, the next follow-up question is, like, do we actually see inflammation in these regions? And what is it about these specific regions of the brain or types of neurons that they would want to participate in fructose metabolism where the rest of the brain does not participate in fructose metabolism? So I currently have a research student here at Central, Kat Giglari, who is picking up on some of those questions, and they are growing some cell culture model of neurons to look at whether or not there's differences in metabolic stress and some other aspects of neural function, and if those are different between neurons that are exposed to fructose versus neurons that are exposed to glucose.
Huh.
Yeah.
That is really
Yeah.
interesting. So let me see if I get this. If the brain is participating in the metabolism of fructose, is that only when we have too much fructose, or no, even if we have, like, small, appropriate amount? I don't know if that's the correct term, but will the brain also participate even in small quantities? Is that it?
Yeah, absolutely. So there are 2 places that our cells will see fructose from. One of them is dietary fructose. So when we eat snacks, so either fruit or things with high fructose corn syrup, or when we're dehydrated, one of the ways that our cells maintain osmotic pressure is by converting a little bit of glucose into this molecule sorbitol, and it is a way of increasing the number of molecules inside a cell so that we don't lose too much water, right? So number 1, stay hydrated. You will reduce stress on your cells and especially on your brain. Number 2, once we've made sorbitol, our body's first goal was to reduce the stress of dehydration to stop us from losing water, but now we have to do something with that sorbitol. And sorbitol gets converted into fructose, and then that fructose has to be metabolized. So we see this with chronic, I don't think that people understand how stressful dehydration is. So when I say chronic dehydration, I don't mean like you haven't drank water in 3 days. I mean like you're letting yourself get dried out over a couple of hours. But if you do this consistently within a day, or like if it's a regular part of your life to not drink water, that counts as chronic dehydration. That is one of the things that contributes to kidney disease. And for us, in our brain, when we have that sorbitol and we have specific regions converting that sorbitol to fructose, there's certain regions of our brain that say, hey, this is a sign of dehydration. So one of the thoughts, and this is something that another paper I was a collaborator on coming out of Rick Johnson's lab in Denver, One of the thoughts is that some of the fructose metabolism in the brain is helping us deal with and sense dehydration. And then, like I mentioned earlier, another thought is that, especially in populations where there was a more temperate or tropical climate where we have seasonal fruits, that fructose metabolism is found in certain regions of the brain that let us know, like, oh, essentially, like, this is harvest time. After harvest comes winter. So when we start eating these fruits and when we see more fructose metabolism, this is probably the perfect time to gain a little bit of weight, because as soon as those fruits go away, what is there going to be to eat? So it's also involved in helping us regulate our response to food availability so that we can survive fasts.
That's really interesting.
Yeah.
It is really
So
interesting.
maybe this might be a question to Kelly as dietitian, but is there, so that means that it's, I think that there's a misconception that it just because it's fruit, so we are able to eat whatever we want just because it's fruit, so it's not unhealthy. And so particularly that's not the case, right?
I would say no, you can't just eat whatever you want. I mean, that's, you know, and I mean, I think.
Even if it's fruit.
Yeah, but I mean, I think even there, if you look at like recommendations, are typically 2 to 4 servings of fruit per day. And I mean, I don't know. I feel like why would you go over that?
So Kelly, I don't know about you, but this is a question that I will get all the time when I talk with metabolism about people, because number one, fruit is healthy.
Sure.
One of the things that is very healthy about fruit is that it has fiber. When we eat dehydrated foods, we don't get any water from that. So natural foods like fruit and vegetables have water in them, and they have a lot of antioxidants.
Yep.
So those are all great. In terms of maintaining our metabolic balance and making sure that we're not disrupting our normal like blood glucose levels or fructose sensing in our intestines and liver and in our brain, if we're not moving around and if we're not going to use those sugars for anything, like for energy production, then we do have to store them somewhere. And for fructose, that's an unregulated pathway. It messes with our ability to properly sense how much we should store. We tend to, if we're taking in a lot of fructose from any source and we're not moving around, then our bodies will scramble to store it in this disorganized fashion. So the way that I'll describe this with students is like, you know, when you come home and you make dishes and then you wash them and you do your laundry and you fold it and you put it away, everything like goes according to plan. And you'll always have some stuff out, but you'll always have, it's not going to be a big mess. If somebody were to just come into your house and drop off like 3 weeks worth of laundry, like it's now a mess. And you might scramble to put it away, and now things are going into the wrong place, and now you can't get to them when you need them. And the same thing can happen in our body when we store fat. If too much sugar comes in and we don't have enough time to store it properly, we'll just end up like stuffing it in all these places where it shouldn't go, and then we have a hard time accessing it later, so we can't turn it into energy as easily.
That's such a great analogy. Loved it.
That's good.
Thank you.
That's really good. And what are the next steps now?
So on the research side, some of the next steps are, like I mentioned, Kat is currently looking at some of the metabolic consequences of fructose metabolism specifically in these neurons. So how this changes different protein functions. I'll leave it at there. If anybody wants to talk more about the biochemistry, you guys are welcome to reach out to me. I love talking about this. But again, this is something that we have some really promising results on the way, so I really hope to be able to share more about that in the near future. So after we have an understanding of what type of stress may or may not be caused by these different metabolic pathways inside a cell, in general, we would like to look at how those cells that have taken on distress communicate with the immune system. And so I did mention Kat's project using neurons. We have some other students in my lab right now doing projects that are looking at liver metabolism, because there's a lot of open questions about non-alcoholic fatty liver disease and how inflammation influences the progression of liver health or disease, and how inflammation also encourages the liver and possibly the brain to become less sensitive to insulin.
Mm-hmm.
So in our liver and in our fat tissue, our adipose tissue, insulin is a growth factor that will encourage those two organs to store glucose as energy for later. Your brain does not really store glycogen and it doesn't store fat, but it is heavily involved in fat metabolism because all of our cell membranes, those are phospholipids. Those are another type of fat. So we are interested in if dysregulating metabolism in the brain changes fat metabolism of our neurons and if that reduces neural function. Because if you don't have enough cell membranes, you can't have your neurons making connections.
Mm-hmm.
So that is like something that a number of different research scientists will collaborate on in the future, not just our lab. But there's
complex.
it's so complex, and there's so many open questions in that. But that's the general direction we would like to go in. We're starting with some of these things that are happening inside the cells, and we would like to start looking at how those cells communicate with cells of the immune system and how cells of the immune system might either sense or respond to the stress of that or of the original cell that's metabolizing the fructose.
That's really cool. So are you recruiting students to work on all of that?
We are not, I am very, very, very lucky that I have a full lab going into next year, so I'm not currently recruiting students, but like you guys mentioned, this is going to be a big project that takes more than one more year, so we are always on the lookout for people who are interested in this. Even if they don't end up working with me and they want to know what it's like to be a biochemist or how to get involved in that, I love helping people find the place that they need to be because it isn't always obvious when we're young, especially like if we don't know somebody else who's currently in this field, like what we're supposed to do or how something got where they were.
Yeah. And now talking a little bit about your career a little bit again, could you maybe share with us some of your academic achievements that maybe you are particularly proud of? That's our job, like highlighting awesome stuff that females do. So, yeah.
Well, I hope this came through when I mentioned some of the projects that my students are working on earlier. But honestly, the thing that I'm the most proud of is getting to work with people at the beginning of their careers and hopefully setting them up for success.
That's really awesome.
Yeah, and I cannot emphasize enough really how proud I am of not just my lab, I'm proud of you guys the most, but like all the students at Central, we have a slightly smaller faculty base that can participate in research. So I think there's been a number of students who I've had in class who don't necessarily realize that research is a career option, or they might not realize that student research is possible at Central. Yeah. So it does mean that the students who are participating in student research did take on a little bit of, they were very proactive in finding these projects and in finding these mentors. And so I always think that it's really amazing to see the projects and the ideas that people are coming with and really being proactive to get started with. And I do have to say, I have been extremely lucky, the people that I've been working with. So Jake O'Hara has been working on the liver project, and he got into a PhD program. So
nice.
He's going to be going to the Washington University at St. Louis next year.
Nice.
And Aaron Valdez is currently finishing up his master's. He's also working on the liver side of the project, and I am just very excited about that. Again, he discovered something that, nobody else has reported on in this aspect of liver metabolism in a specific genetic mutation that people have. So humans can have this genetic mutation called hereditary fructose intolerance. Mm-hmm. One of the mouse models that we use is a genetic mimic of that. So the mice also have hereditary fructose intolerance. And in Aaron's characterization of some of those livers, he was like, oh, hey, there's this difference that we're going to publish soon, so I'm going to leave this at spoilers, but it was really fantastic to have somebody come in and point out something that nobody else has pointed out before and get to work with them through that time. Be like, no,
that's
you're not.
Cool.
Yeah, you're not just some student here at Central. You've discovered something that nobody else has seen before. And, you know, I could just go on about that. But yeah, so the things that I really am the most proud of are the work that I've done with, you know, collaborators who helped train me, like Dr. Tolina at Boston University, and then the people that I have been able to train as we just hopefully like contribute to our understanding of how bodies work.
Yeah.
Definitely. So for students interested in following a similar career path, what advice do you give these students?
Yeah, I think that one of the things that is super helpful is there's a difference between an area you're interested in intellectually and what you enjoy doing on a daily basis. So I think that if someone is interested in health or in science, you know, be it like nutrition or like clinical trials or biochemistry, talk to somebody who's currently doing that, and then just ask them what their like day-to-day life was like when they got started, because there are so many different ways to be involved in science and in health and in medicine, and you should not only just like the area you're working in, but you should also like what you're doing on a daily basis.
For sure.
Yeah.
So important.
Yeah. So I think that, you know, talking to people who are doing that is super helpful. And then I also think that as we go through our careers, finding a mentor that you get along with, and really more than one mentor, is super important, because you can love a certain topic and you really need somebody who like is invested in you personally,
Right.
to help you get started, because like getting your master's, getting your PhD, it's a long trial, and you have to really like what you're doing. It does pay off.
Yeah.
It definitely pays off. But it takes a long time. And, you know, even if somebody didn't go on to graduate school, just finding those career mentors who know you and can give you advice and guidance that is going to take into consideration like who you are as a person and the resources you personally have to go after something, can be the difference between seeing your path forward versus seeing obstacles and not quite knowing how to overcome them, if that makes sense.
Yeah, for sure.
Yeah.
I think a
That's awesome.
bad mentor can also ruin a passion for someone. I mean, I've heard that before, unfortunately, and so I think that's really important.
Yeah, I don't. You never want to be negative.
No, I'm not trying to be negative.
Yeah, no, but I mean, I think like we've all,
happens.
it does happen. It happens, and I think so Kelly, I'll just go on this slightly longer tangent, but my first job out of my PhD program, I was super excited about, and I didn't find a great mentor there. There was nothing wrong with anybody there.
Yeah.
And it was a great company, and I will not give more details about that right now, but I didn't ever find like a good mentor there, and it became obvious that I was going to, if I tried to stay there, I would just burn out. I'm like bitter about being a scientist. But it was very hard to finish my PhD and land this job where everybody was like, oh my God, you did it. Like, this is it. And I really thought that I really did. I was like, oh my gosh. And then when I was just like, yeah, I have to leave, it was very personal, because I felt like, oh, did I fail as a scientist because I'm not working out in this specific role? And I was lucky enough to talk with somebody that I went to grad school with who was further along in their career, and they're like, yeah, it's not you. And at first I was like, okay, this person is my friend, you know, of course they're going to say that. And then a couple of years down the line, they were working in another company, and they were hiring for a team, and they're like, hey, we need a biochemist, like, I need you to come in and chat with somebody. And that was it was super validating, because I'm like, oh, you didn't say that, like, you actually thought I was a good scientist. And sometimes when we're, at least for me, when I was younger, running into a bad mentor, it was very hard to figure out, well, like, am I just not trying hard enough or anything like that?
Right.
But it is
hard to know.
Yeah.
It is hard to know.
Uh-huh.
But that is the
true.
truth of it.
It is.
You do need somebody to be a good mentor. And if you haven't found that in the person you're working directly with, don't take it personally.
Right. Right.
Like, obviously be self-reflective, but don't take it personally, and find yourself somebody who is an advocate and just talk to a number of different people, because we all do need something different. Every situation is different.
Yeah, totally.
That's awesome. So before we wrap up, can you provide any contact information that people can follow you? I don't know, social media, or how people can find you?
Yeah, absolutely. You can usually find me in the lab, or if I'm not there, I'm like running around. Washington State is absolutely beautiful, and there's a lot of different places you can go trail running. And then on social media, I do try and write about metabolism for a more general audience. And so askabiochemist.com is a blog, and then I am also on Instagram at askabiochemist. And I don't do other social media because I find it overwhelming.
As we all do.
It's too much. All right. To our listeners, if you found this episode as engaging and insightful as we did, please subscribe, leave a review, and share it with your friends, family, and colleagues. Follow Science Her Way on social media and YouTube. Stay tuned for more remarkable stories. Thanks for tuning in.
Thank you so much, Dr. Sarah, for coming, and we really appreciate your time. This was awesome. Thank you.
Yeah, thank you guys. It was so much fun to talk about it. And again, if anybody else is interested in metabolism or biochemistry, I'm willing to keep talking about it.
Okay, guys, stay tuned, and thank you for listening.